Hello students. Now I'm going to discuss about modified TI plasmid retrovirus, the steps of recombinant DNA technology, etc. So now start with modified TI plasmid is a non-pathogenic but able to deliver desirable genes into plants. So modified TI plasmid is non-pathogenic but able to deliver desirable genes into plant. Modified TI plasmid is non-pathogenic but able to deliver desirable genes into plant. Next, retrovirus. Retrovirus can transform normal animal cells into cancerous cells by chopping the disease-producing DNA segment. These retroviruses are modified and are used to deliver genes of our interest into animal cells. So, it can transform normal animal cells into cancerous cells by chopping of disease producing DNA chopping of disease producing DNA segment these retroviruses are modified and are used to deliver genes of our interest into animal cells. Okay, now next is process of recombinant DNA technology. Process of recombinant DNA technology. Okay. Recombinant DNA technology involves several steps in the following sequence. Number 1, isolation of genetic material such as DNA. Separation of isolated DNA fragments. Next, cutting segments of isolated DNA at specific sites by restriction enzymes. Next, amplification of genes of interest using PCR. Inserted and ligation of DNA segment into vector and formation of recombinant DNA. Insertion of recombinant DNA into host cell, that is transformation of host cell. Next, selection of transformed cells. Next, culturing transformed host cells to obtain clones, that is cloning of recombinant DNA or culturing transformed host cells to obtain desired product at industrial level. So, recombinant DNA technology involves recombinant DNA technology involves several steps in the following sequence. Number 1, isolation of genetic material or DNA. Isolation of genetic material or DNA. Separation of isolated DNA fragment. Separation of isolated DNA fragments. Next is cutting segments of isolated DNA at specific sites by restriction enzymes. At specific sites by restriction enzymes. Next, amplification of genes of interest using PCR. Amplification amplification of genes of interest using PCR. Number 5 is insertion and ligation of DNA segment. Insertion and ligation of DNA segment into vector and formation of recombinant DNA into vector and formation of recombinant DNA. Okay. Next step is insertion of recombinant DNA into host cell, that is transformation of host cell. Insertion of recombinant DNA into host cell, that is transformation of host cell, that is transformation of host cell. Next step, selection of transformed cells. Selection of transformed cells. Next, cutting transformed host cells. Culturing transformed host cells to obtain clones. That is cloning of recombinant DNA. That is cloning of recombinant DNA. And the last step is culturing transformed host cells to obtain desired products, or culturing transformed host cells to obtain desired product at industrial level. Okay. Now next is bioreactors or fermenters. Okay. Next is what? Bioreactors. Bioreactors or fermenters. Now, what is it? For large scale production, for large scale production or commercial production of bioengineered products or recombinant proteins, bioreactors are used. These are also called fermenters. So, for large scale production, for large scale production, or commercial production, of bioengineered products or recombinant proteins, bioengineered products or recombinant proteins. Bioreactors are used. Bioreactors are used. These are also called fermenters. Okay. Bioreactors are large vessels in which raw materials are biotechnologically converted into specific products. Vessels in which raw materials are biologically converted into specific products using large volumes of 100 to 1000 liters 1000 liters of culture of microbes or plants animals or human cells, plants, animals or human cells. Okay. These have adequate arrangement for adhesion. These have adequate arrangement for adhesion. Agitation agitation, drain or overflow vent to remove the waste biomass. Drain or overflow, drain or overflow vent to remove the waste biomass along with their products, okay. Optimum growth condition like temperature, pH, oxygen, vitamin, nutrients and substrate are maintained inside the reactors for obtaining desired product at the maximum rate. Optimum growth conditions like temperature, pH, oxygen, vitamins, nutrients and substrate are maintained inside the reactor for obtaining desired product. At the maximum rate. Okay. In my next video, I will discuss about downstream process and application of recombinant DNA technology. So now, bye students.